So I'm here today to give you folks a break from talking about tear film. I will be talking about functional markers as opposed to molecular mechanisms. We're going to be looking at vision, ocular pressure, and corneal vascularization, and the effects of Far-UVC. In case my title is too subtle for you all. The bottom line of today's talk is that chronic Far-UVC doesn't affect vision or the functional marker measures in our study. I'm the 45th speaker here, so your attention may be running thin. So the reasons you should tune in here, this is a study looking at chronic exposure, which is of particular interest given we are advocating for use of Far-UVC in a chronic setting. The functional measures we're going to be looking at are not dependent on any particular molecular endpoints, so if the mechanism of cellular damage is not CPDs, it could still be captured within these measures. And people really care about their eyes, as proven by the fact that you're all here this morning. So for this study, we use the SKH1 mouse. This is a bald and immunocompetent mouse that has been used in skin research pretty extensively. It is an outbred line. Its genealogy is unclear, and its vision has not been previously assessed as far as we know. The reason we use this line is because this study was originally aimed at looking at skin findings, but we figured, why build a whole other cohort when we could just use the chronically exposed eyes we already have. As for the exposure, you can find more details on this in our previous publication, but it was a 66-week exposure, 8 hours per day, 5 days a week. We used KR, CL, etc. microprotoplasma lamps with filters, as you've seen many times. We had 12 males and 12 females in each condition, with conditions ranging from high with a 400 millijoule per centimeter square exposure all the way down to an unexposed cohort. And you can see the heat maps here, again published previously, as well as the configuration of our cages up here in the upper right-hand corner. You will notice some differences in the heat map from the front to the back setting. We did rotate the cages between the two, and the dosages here are the averages. So, if you've ever tried to give a mouse an eye exam, you might have found yourself in quite a difficult situation. We had to get clever. The two markers we're going to be talking about for vision are acuity. You're likely familiar with this if you've ever gone to the eye doctor and looked at a snowing eye chart. The smaller letters require a higher acuity. And then contrast, which you can see here. This is an Arden grid, no relation, but it is a diagram that looks at both acuity on the x-axis as well as contrast on the y-axis. And greater contrast sensitivity means a greater ability to gauge a smaller gap between two different colors. So the way we did decide to assess this was optokinetics. This is an approach that has been used in previous studies. I think it's an underutilized approach. The idea essentially is to put a mouse on a platform, as you can see up here, and you simulate a virtual cylinder around the mouse. The lines are adjusted based on the position of the mouse's head to maintain the correct distance between them, and the field lines, as they're called, can be made closer together or further apart, closer together representing and requiring a higher acuity, and the lines can also be made more gray to test contrast sensitivity. And essentially the idea is you look for an involuntary response known as tracking, which mice will do when they first notice a moving object. Unfortunately, my video doesn't appear to be playing. Disappointing, but it is a very cute mouse which very subtly follows lines moving either clockwise or counterclockwise, and you can track based on that tracking or lack thereof if they're able to see the field lines at the current settings. So let's just jump into results. So we're going to do acuity first. So I'll give you a moment to get oriented to There we go. So this is our acuity result. So we have in blue the male mice, in red female mice, and then in black that is both aggregated together. You can see conditions along the x-axis. The first finding that we were rather surprised by was that female mice had better acuity than male mice, and that across all conditions there was not a significant difference in female, males, or in both aggregated in terms of visual acuity. One other note on this slide is that the mice tested in this study, the SKH1 mice, did perform significantly worse than mice in previous studies have. They were generally around 0.4 cycles per degree versus our mice averaging around 0.25, a little bit below. The next result is contrast. This one is a somewhat more complex picture, but you'll see a similar result. The different conditions tend to cluster around each other, but in the medium cohort, due to time constraints and the fact that the mice would be nearing the end of their lifespan, and we wanted to have a significant sample size for the next steps that I'll be showing you, we were not able to do contrast sensitivity for the females. We did note, as with acuity, contrast sensitivity is better in female mice than in male mice. And while we initially had a significant difference with lower visual contrast sensitivity in the median condition, when we stratified based on sex and reran the analysis, we found that significant difference vanished, meaning it was a prime example of the effects of confounding. I will note here as well, the contrast sensitivity in this mouse line is lower than what has been noted in previous studies. So now moving on to some of the other measures. Ocular pressure, as measured by tonometry. Ocular pressure can be elevated in conditions such as glaucoma, leading to blindness. And when we assessed it in these mice, we found a similar unextraordinary result. But here there was not even a sex difference. We found that pretty consistently across the board, there was no alteration in sex based on sex or based on condition. And here's the aggregate at the end based on sex. The next marker we wanted to look at, as was discussed earlier this morning, was corneal vascularization. I won't go into it because I think it's been discussed very effectively already. These mice did have pretty extensive corneal vascularization. Our approach to do this systematically was to take visual captures of the mice while conscious and then skeletonize the image. And from the skeletonized image, we were able to calculate the percentage of area that could be analyzed, as in it was in focus, and the total percentage of area covered by vascularization was calculated. So with those results, we found, again, similarly consistent levels of corneal vascularization, both across sexes as well as across exposure conditions. So, just one more example of a null result. In summary, across the board, we did not see an effect of chronic exposure, even in very high doses, 400 millijoules per centimeter squared. We did see an effect of sex on visual endpoints, which explained the one significant difference we had due to confounding. We've concluded that the SKH1 mouse line does seem to have some visual effects at the baseline, which may be related to the corneal vascularization, which is known to be a potential cause of reduced vision. So you can look at this one of two ways. You can look at this as a negative result. It would have been really nice to have some marker for elevated Far-UVC exposure, such as a minor rise in intraocular pressure that we can easily gauge. Unfortunately, we don't have anything like that. But you can also look at this in a much more positive light, which is even with 66 weeks, 5 days a week, 8 hours a day, at exposures that seem to break the occupational limit, we did not see a meaningful detriment to vision. So with that said, there were a couple limitations in the study. We did assess conscious animals. That did make it somewhat difficult to capture eyes completely, so we were only able to do partial areas and not on every animal. The animals were receiving Far-UVC from above, and it's somewhat difficult to gauge exactly how much incident light they actually got onto their eyes as opposed to any other part of their body. Again, they also huddle with each other. And week to week there was some unevenness in exposure. Again, you can read about that in our first publication. Additionally, the SKH1 line is not terribly well characterized in terms of its ocular activity. And we didn't have a good marker for oncogenicity, which when I speak with folks anecdotally about our research is something people are concerned about. When I spoke to one of my attendings about our research, the first thing he said was, "That sounds carcinogenic." Love to be able to dispel that. So, with that said, I'd like to just credit everyone in the planning lab who worked out the study. This is a picture from the Met Gala. I'd also like to thank our collaborators at the Brenner Lab. I don't have a picture of them, so here's a picture of my cat, Studio Chicken. And I welcome any questions you may have.